If you guys have any doubts with the course, you can write it in the chat box. Or I think you guys should be able to unmute yourself. If you want to ask, you can ask. I hope I'm audible. Can one of you confirm?
Yes, ma'am.
Yeah, hello.
Yeah. I
Ma'am, what do we practice numericals problems? Solar Karki or else?
I'm sorry, I didn't get you.
Ma'am, when do we practice numerical problems? Solar Karki or any other books?
You can choose any book. It doesn't matter. Most of the books will have the same sort of problems. Whichever is available to you, you can use that.
Okay, ma'am.
Yeah.
Thank you.
What's your subject in MSc?
I'm sorry, I didn't hear you.
What subject? Your MSc, ma'am. Your MSc subject, ma'am.
Uh,
What's your MSc subject?
yeah, I did botany.
Okay, ma'am.
I think 2 more minutes.
I want to
Yeah.
do my MSc in biotechnology, ma'am. Can you please tell me some courses, ma'am, to add them in my resume,
Um,
which are available on the website, ma'am?
a lot of courses for MSc are available. It depends on what sort of uh, interests you have.
Biotech, ma'am. MSc Bio.
Yeah,
I want to have
biotech.
my MSc.
Yeah, so a lot
Yes.
of courses are there. Like this bioreactor course was there.
Okay,
There
ma'am.
were courses on cell biology, molecular biology. All
Okay, ma'am.
these are included in the biotech field itself. It's a very broad field, right? So,
Mm,
depending on maybe if you want to go further in research, depending on maybe
mm.
what sort of research, or if you want to go in an industry position after your MSc, maybe
Okay,
depending
ma'am.
on that, you can choose your subjects.
Okay, ma'am. Thank you, ma'am.
Hmm. Yeah, I think it's 6:05. I think we can start. I'll explain how this will work. So, because NPTEL courses usually don't have interactions, that is why we have come up with this sort of format in which every week we'll have one live session and we can clear doubts or talk about any problem that anyone is facing or ideas or references. Okay. So each week, this week I'll be focusing on whatever was taught in the week one portion of the course of bioreactors. Okay. So we can either start by you asking me questions about doubts that you have, or we can directly. I have like 10 questions that are similar to the ones that are there in your assignment, and we can solve them. Since assignment 1 has mostly very direct questions, it won't take a lot of time to solve these 10 questions either. So, do you guys have any doubts? I can't solve the current assignment with you, that will be cheating, but I can explain maybe how you can do it if it is a numerical problem. Does anyone have any doubts regarding any of the three assignments? There were assignment for week 0, assignment for week 1, and a practice assignment too, right? Does anyone have any doubts for that? Abhinav, if you're saying something, I can't hear you.
No, ma'am, nothing.
Yeah, all right. So, no one has any doubts. Were you guys able to solve the problem? Have you attempted the assignment 1?
Yeah, yes, ma'am.
Yes, ma'am.
All right. Did you guys have problems with the assignment 0, which was mostly maths?
Yeah, yes, ma'am. That was
Which,
mostly maths, ma'am.
yeah, did you have a problem with it? Were you not able to solve something? Uh, okay, then I'm assuming everyone has the prerequisite knowledge of maths. Yeah, and you didn't find that tough. Okay. So if none of you have any problems with the assignments till now, maybe we'll go ahead and solve a few other problems. It won't be numericals. We still haven't gone much into detail to give numericals. It will be mostly direct questions like you've seen in um, your assignment 1. So I'll just present my PPT. Yeah. I hope my PPT is visible to you, right?
Yes, ma'am.
Yeah.
Yes, ma'am.
Thank you. So, before I start, I just wanted to go over the topics that you have been taught this week. So, first of all, Sir has told you about what are the needs for a bioprocess, right? He explained how we make monoclonal antibodies, or how we get biofuels, or how even curd is technically a bioprocess, right? And then he went on to explain the different kinds of bioreactors that you can use and the modes of operation, may it be batch, fed batch, or continuous. And in the second video, someone is joining. Is my screen moving forward?
Yes, ma'am.
Yeah, all right.
It's in the second slide, ma'am.
Yeah,
It's in the second slide, ma'am.
yeah, it's in the second slide. Correct. Okay. So, in the second video, we briefly talked about the modes of sterilization, and then we did a few numerical sort of derivations through which the end was we wanted to calculate the decimal reduction point, right? Which is the amount of time it takes for the amount of uh, cells to reduce to 10%, right? So let's go to the first question. So, hybridomas are, and the option are antibody producing cells in the body, cancerous cells in the body, a fusion of antibody producing cells and cancerous cells, or cells producing monoclonal antibodies. I just want to tell you that few of these questions may have multiple answers. So, does anyone want to answer? Or if you want, you can put the answer in the chat box too.
Single type of antibody.
Answer is C. Okay. Poonam thinks the answer is C. A, B, C, D. Okay. D, C.
No, I think even C, both C and D may be the correct numbers.
As people are still joining. Okay. A lot of answers are there. C, mostly C's, some D's. A, C, D. So before I go ahead, I just want to explain in brief what hybridomas are. Okay, that will make it easier for you to understand what the answer is. So hybridomas are cells which produce only one type of antibody. Now all the B antibodies are produced by B cells in our body, right?
Yes, ma'am.
Yeah. But those B cells, the thing is, they don't have a long lifespan. So if you passage them, which is if you keep culturing them maybe a few times, they will stop growing, which is not what we want. We want an infinite source of the antibodies for our use, right? So what we want is something, a cell which is able to propagate continuously and to produce antibodies. So how hybridomas are formed is by this method. So um, antibodies are produced against antigen, right? So there is something that the
Yes.
antibody will recognize. So what we do is we take a solution of that antibody and we inject it into the mice, which is called immunization of the mice. Now, since this is a foreign body to the mice, what it will do is it will produce B cells against these antigens. And these B cells will be able to produce antibodies against these antigens. Okay, so we will take the spleen of the mice after it has been immunized, and from that we will collect the B cells. Now, these B cells will not just be against your antigen, it will be against many antigens that the mouse has faced. But like I said, these B cells don't have a long lifespan. So, what we do is we take myeloma cells, which are cancerous B cells.
Yes, yes.
Okay, so cancerous cells have the property to be able to grow continuously. Okay, they don't have a passage time. So you can cancer cells, if you culture them, they will keep growing continuously for how much ever long you want. So you fuse both of these cells. Okay. And once you fuse them, you have a cell which is immortal but also capable of producing these antibodies. Okay. So that is what a hybridoma cell is. Okay. And from the many hybridomas that will be formed, you will isolate the hybridoma which will react against your antigen, and that you will use to produce many antibodies that you want. Monoclonal antibodies means antibodies which are against one epitope. Okay, so in this case, the answer would be both C and D. Hybridomas are a fusion of antibody producing cells and cancerous cells because they're a fusion of B cells and cancerous cells, and they are also cells which produce monoclonal antibodies. So those who said C and D are correct. Does anyone have a doubt in this question? All right, I'm assuming no. Okay, so let's go to the next question. Okay, so this sir has talked about in his lecture. So microalgae is used in the production of biodiesel as, and the options are microalgae is photosynthetic, microalgae accumulates bio-oil, microalgae can be heterotrophic, and microalgae cultures do not require pH control. So this time, what do you think the answer is?
Option B, ma'am.
Option B. Anyone else has a different answer? All right. The answer is actually B, because microalgae accumulates bio-oil. We don't use microalgae is photosynthetic, but that is not the reason that we propagate them to produce biodiesel. It's because they accumulate bio-oils. All right. So let's move on to question number 3. So in this, we have to match the following components of bioprocessing. So in A, you have C. glutamicum, which is a bacteria. sugarcane bagasse and glutamic acid, and in B you have substrate, product, and inoculum. So which one do you think is matching?
For option 4.
Option 4. Anyone else has a different answer? Now I'll just explain what each of these terms means. Maybe that will make it easier. So substrate is the media or whatever feed you are giving to the cells or the enzyme, which will be converted to your product. And product is what the final product you desire. Inoculum is maybe enzyme or cells that will convert the substrate to the product. Does it make that easier? There's only one correct answer to this question. And the answer is D. Yeah. So C glutamicum is the inoculum because that is what is converting the substrate to the product. Um, Sugarcane bagasse is the substrate because that is what this bacteria is feeding on, and glutamic acid is the product that you get because this bacteria will feed on sugarcane bagasse and release glutamic acid, which we will extract and use for our needs. All right. So if no one has any doubts in this, I'll move on to the fourth question.
Okay.
Alright. So from below, identify the type of bioreactor where aeration and agitation is brought about by pumped air alone. And the options are stir tank bioreactor, airlift bioreactor, packed bed bioreactor, and solid state bioreactor. I'll just explain what aeration and agitation means in this.
Option B.
Okay. Aeration means introducing oxygen or air into the media, and agitation is proper mixing of the media so that settling doesn't happen. So, what do you think the answer is?
Airlift bioreactor.
Okay. Anyone else has any different answer? A. Okay. All right. The answer is actually B. It will be the airlift bioreactor, and you will be able to see in these diagrams. So, in this case, in the stir tank bioreactor, air is in fact given through a sparger, but the mixing is done using an impeller. There is a mechanical mixing happening. In packed bed bioreactor, there is no mechanical mixing happening, but there is an air inlet. And same in the solid state bioreactor, you have an impeller and an inlet. Okay, but only in the ALF bioreactor you have a sparger, which is the um, portion of the bioreactor through which gas is put into the media, and that is what causes the mixing of the media in this case. So the answer is in fact airlift bioreactor. Okay. If no one has a doubt with this question, I'll move on to question number 5. Okay. So to carry out plant tissue culture, a medium with all essential vitamins need to be prepared. And does anyone have any doubts? I just heard someone.
No.
All right.
No.
Okay. So yeah, these components are heat sensitive because they are vitamins, right? So from the below options, choose the best option to sterilize these components. And the options are autoclaving, chemical treatment, membrane filtration, and high pressure application. So in this
It's
case,
B and C.
B and C. All right. Anyone else has any other B and C? Okay. Any other answers? C. Okay. B and C. Okay. The answer is actually just C. The thing is, you can't autoclave it, which will be heating it at high pressure because these vitamins will get degraded and they won't be of use anymore, right? And chemical treatment has a similar problem. Chemical treatment will change the chemical structure of the vitamins itself, and then they also won't be of purpose. High pressure application by itself has no sterilization properties. So the answer is membrane filtration. So in this case, the membrane will have very, very small pores through which your media can pass. But bacteria and virus will not be able to pass because the pores are smaller than the size of the bacteria and the media. Right. Again, I'm moving on to question number 6. Okay. So the question is identify the mass balance equation, and mass balance in this case means where both sides of the reaction the mass has to be equal, right? For a microbial uh, for microbial cells in a bioreactor under batch operation. Okay, where Ri is the rate of input of cells, Ro is the rate of output of cells, Rg is the rate of generation of cells, and Rc is the rate of consumption or the rate of death of cells. Okay, and dA by dt is the rate of accumulation of cells. So what will be the mass balance equation?
Third option.
Third option. Okay. Anyone thinks it's some other option? Do you think there's a different answer? C. Okay. Most of you seem to think it's C itself, right? So before I move on, I just want to remind you what the different modes of operation is. First is the batch operation in which all the nutrients are added at one point at the start and till the end we don't add or remove anything from the reaction. Fed batch is a modification of batch fermentation in which nutrients are added at intervals depending on the need of the culture, and continuous mode of operation is which fresh medium is added and spent medium is removed continuously. And in this case, since it's a batch reaction, that means there is no input of cells and there is no output of cells. There is only generation of cells, that is the cells which are dividing, and any death of cells. So the option is correct, it is C. Right? So again, I'm moving on to the next question. Just stop me in case you have doubts with any of the questions. Okay, so during thermal sterilization, the logarithm of viable cell concentration percentage is directly proportional to this. Sir has talked about in his videos. So which answer do you think it is? The options are A is the initial pH of the medium, B is the time of exposure at high temperature, C is the dissolved oxygen, and D is the temperature of sterilization.
Option B.
Option B, okay.
Yeah.
Okay. Any other answer? B. Alright. You guys seem to think it is B, and that is in fact correct because the percentage of viable cell concentration is directly proportional to the time of exposure at the set high temperature. Alright. Okay, so let's move on to question number 8. So identify from the below expression denoting the decrease in the rate of the viable cell concentration RD during thermal sterilization of a solution of well dispersed cells, given that the viable cell concentration present at any time is xv. Again, sir has talked about this in his videos. So what do you think the answer is? A. Okay.
Yes, we will.
All right. Okay. Answer according to the chat are A, and yes, that is in fact correct because we know that the rate of decrease in the concentration of viable cells is directly proportional to the concentration of cell at the time, which is xv, and kd is just the constant. Okay. And A was the correct option. Okay, so question number 9. In a certain bioprocess involving a very high value product, the microbial cell has a very low conversion of substrate to product rate. That means that no matter how much substrate you put, the rate at which the microbe will convert the substrate to the product is very low. Okay, and the accumulation of the product also causes a reduction in this conversion rate. That means there is a negative feedback. So the more the product is there, the less the microbial cells will produce new product. Okay. So which of the following operation modes is best for this bioprocess to achieve the efficient conversion? So option A is continuous operation alone. Option B is batch operation alone. Option C is fed batch operation with product removal alone. And option D is both continuous and fed batch operation.
Option D.
Yeah. Okay. Any other answer? D. Okay. Then it is in fact D. So with both continuous and fed batch operation, you get to remove the product. Okay, because you're removing the product, and since you're adding new media in both continuous and fed batch, you get the microbes will be able to convert substrate to product again. Okay, this will not happen in batch operation because at one point the substrate concentration will be so high that the cells will not the product concentration will be so high that the cells will not convert any more substrate to the product. So if you want to get a high product rate, you can use either a continuous or a fed batch operation in which the product is also being removed. And the final question is a sort of numerical. So um, this also sir has explained a different sort of question, but very similar to this. So the question is the media used for a bioprocess is contaminated with a single cell, I mean with single cells, similar kinds of cells with similar thermal response characteristics. Okay, and it has been observed by maintaining the media at 60 degrees Celsius for 15 minutes. 65% of the cells become non-viable. So you have to determine the decimal reduction time. So choose the closest answer to your calculated answer. Okay. So I'll give you guys maybe 5 minutes and then you can write the answer in the chat box. What you are getting. Okay. Grab a pen and paper and try to solve this.
I'm at 33 minutes.
How much?
33 minutes.
All right. Do you want to maybe explain how you solved it so that others can also get to know in case they were not able to?
Ma'am, first we need to find out the KD value, that is the death rate.
Yes.
Then after finding it, we will use the decimal reduction time, which means only 10% of the cell will be living. So it will be something like KD is equals to 1 by time it takes to reach that 10% in ln 10 by the time it takes.
Okay. But is it
After finding the
10%?
KD, we'll use it to find the time of decimal reduction time.
Yeah, but when you're finding the KD, are you saying that 10%? Where are you getting that
No,
10% from?
10% is for the second part when we have to find the decimal reduction. First percent is only 35% is the final concentration.
Yeah. Okay. Is everyone getting answer A, or is anyone getting any other answer, or a completely different answer which is not there in this? Alright. So maybe you guys have not solved it. Should I give you guys more time to solve it, or should I sort of explain how this is done just once? Anyone who wants more time to solve this question? A. Okay. Subhashri saying A. Okay. So, okay. I'll just explain once how it is done. So, from what Sir has taught us, we know the formulas for the decimal reduction point, which is D equals to 2.303 by KD. But in this case, we don't know what KD is. Okay. And like Sundar just told us, we know how to find KD. There is another formula for time. Um, and if we substitute the values, if we rearrange the formula in the first place, so that what we're trying to find is KD, and we substitute whatever information is given to us in this, we will get the KD. So time is given as 15 minutes. If you convert that into seconds, it will be 900 seconds, right? And you know the initial concentration is 100%. And since At after 15 minutes, 65% is reduced. So 100 minus 65, what is remaining is 35% of the cells. So to get KD, you have to input the time, which is 900, x v naught, which is 100, and xv, which is 35, and KD you will get as 0.00117 about. Okay. Now that you've gotten KD, you can find the decimal reduction point, which will be 2.303 divided by this 0.00117. So you will get your answers in seconds, which will be 1968 or 69 seconds. If you divide that by 60, you will get 32.80 minutes approx. Okay, and the closest answer to that will be this 32.90 minutes. You might get a slightly different answer because all of us are calculating a little differently, but the answer should be close to 32.9. Okay. Does anyone have a doubt with this question? Uh, Subha, did you get what I explained? Should I explain once more?
Excuse me, ma'am.
Yeah.
Ma'am, even in the week one assignment, we have a question where we are getting the answer approximately 18.1 or something.
Okay.
But in the options, it is the nearest answer it is given is something like 17.85. So we can choose that option, right?
Yes, sure.
It's close.
Yeah. Because depending on how many decimals you take or what calculator you use, if you're doing it with hand, you might end up getting a different answer. But it should be the
Yes.
closest to that answer.
Okay. Thank
Yeah,
you.
yeah. Okay. Subha, were you able to understand when I explained it? Should I explain it once more? You can just type in the chat box. Understand? Okay, sure. Anyone else who didn't understand how to solve this problem, because it's there in your assignment and it might just come in your end final exam. Yeah, all right. Okay. Uh, then I'm assuming all of you have understood. This PPT will be shared to you, and this recording also will be available to you. Um, but for today, this is all the questions that I have prepared, and these are all very similar to the ones that are there in your assignment. And most of the questions are very direct answers from the video. If you watch the video, you will get the answer, right? And only maybe one question like this is a numerical. So apart from these questions, does anyone have any doubts with assignment 0, practice assignment, or assignment 1? I can't help you with, but if you have a doubt, I can maybe try to explain something.
No problems.
No, I'm sorry, I couldn't hear you.
No, ma'am. No problems, ma'am.
No problem. Right. Does no one else have any problem? Is there something that you will like for me to explain in the next session? If
Ma'am,
you,
one more thing.
yeah,
Ma'am, the assignment for week two, which is the second quiz,
yeah,
will it be coming next week or by the end of this month itself?
by next week it will come. I
Okay.
mean, yeah, next week.
Okay.
Yeah, don't worry about it. I think the deadlines are pretty generous, right? You have a good deadline to solve these questions, right?
Yes, ma'am.
Yeah, so I don't think it should be a problem. Um, if you have any doubts, there is also a discussion forum regarding this um, live session that we had, or regarding any of the course videos or assignments. You can put it in that discussion forum also. Um, if you guys don't have any doubts, you can leave the meeting. If you have any doubts, you can stay. If you have any suggestions for something you want to be done for the next session, you can tell me.
Ma'am, do we have a live class every week, ma'am?
Yeah, you have.
Okay, ma'am.
Yeah.
Thank you, ma'am.
Yeah, no problem. I'll mostly be discussing that week's assignment so that it's easier for you to solve it. But
Okay, ma'am.
apart from that, also, if you want clarification about something that was taught in the video and you've not been able to understand it, you can ask me and I'll explain that too.
Sure.
All right.
Yeah, thank you, ma'am.
Yeah, no problem. Thank you for attending.
Bye, ma'am.
Bye.
Thank you, ma'am.
Thank you. Someone raised a hand. Do you have a doubt? Just a mistake. All right. Thank you, guys. See you next week.